Imagine identical twins, same DNA down to the letter. Same face, same height, same eye color, same everything your genes are supposed to control. Now press both of their thumbs onto an ink pad. The two prints will be completely different, not similar, not close. A trained examiner could separate one twin from the other using nothing but those 10 fingertips, with total confidence every single time. That should not be possible. If a trait is written into your DNA, identical twins should share it perfectly. That's the whole premise of identical twins. But fingerprints break the rule completely, and they're one of the only physical traits in the human body that do. So where does the difference actually come from, if not from the genes? The story of fingerprints goes back further than DNA itself. In the late 1800s, the British scientist Francis Galton became obsessed with proving that fingerprints were both unique to each person and permanent for life. He spent years collecting and comparing prints, including from twins and multiple generations of the same family, trying to find any genetic pattern in them. His work eventually became the foundation of modern forensic fingerprinting, used by police forces within his own lifetime to identify suspects. Galton could prove uniqueness, but he could not explain it. He had no idea what was actually drawing those tiny ridged lines onto a fetus's biotip in the first place, and he said so plainly in his own writing. For over a century after Galton, the actual mechanism stayed a genuine mystery. Scientists knew fingerprints formed before birth. They knew the pattern never changed afterward. What they didn't know was what process inside the womb was responsible for laying down ridges in loops, swirls, and arches, instead of, say, a perfectly smooth fingertip or some random scatter of bumps. Here's the detail that makes it stranger. Fingerprints start forming around week 13 of pregnancy, and the basic ridge pattern is fully locked in by around week 24. That's an 11-week window deep in the womb before a fetus has done almost anything except exist and grow. Whatever decides your specific pattern has to happen entirely inside that narrow window, with no input from the outside world at all. The real answer didn't arrive until 2023, when developmental biologist James Glover and his team at the University of Edinburgh finally mapped the actual mechanism in a study published in the journal Cell. And the mechanism turns out to be a fight between three molecular signals, not a blueprint. One signal called WNT tells skin cells to multiply and start forming a ridge. A second signal, EDAR, gets switched on by WNT and then amplifies it further, pushing the ridge to grow more. A third signal, BMP, does the opposite, actively suppressing ridge formation wherever it's active. Where WNT and EDAR win, you get a ridge. Where BMP wins, you get a flat valley between ridges. The two sides essentially fight it out across the surface of the fingertip. Glover's team found that this fight doesn't happen all at once across the whole finger. It starts at several separate points and spreads outward as waves, like ripples on a pond, except multiple ripples are starting at once from different spots and slamming into each other. Where two waves meet head-on, you get a straight line. Where one wave curls around another, you get a whirl. Where a wave hits an edge and bends, you get a loop. The final fingerprint is just a frozen record of exactly where and how those competing chemical waves happen to collide. This wasn't just a theory built from observation. To actually prove it, Glover's team tested the mechanism directly in mice, which don't have fingerprints on their fingers, but do have similar striped ridges on their toe pads, built from the same molecular system. When the researchers artificially boosted or blocked WNT, EDAR, and BMP signaling in the mice, the ridge patterns on their toes changed in exactly the way the model predicted. More ridges, fewer ridges, different spacing, on command. That's the difference between a guess and a mechanism. They weren't just describing what happens, they could reach in and change it. That single mechanism explains everything that genetics alone couldn't. A person's overall finger size and ridge spacing is still set by their genes, but the precise starting points of those chemical waves are decided by tiny, essentially random local conditions. Even in identical twins sharing a womb, the exact pressure of amniotic fluid against each fingertip, the position of each twin's hand against the uterine wall, the density of fluid moment to moment, all differ slightly between the two fetuses. None of that is written in DNA. All of it nudges exactly where each wave starts and how far it travels before colliding with another. So fingerprints aren't really a genetic trait at all, not in the way your eye color is. They're closer to a fingerprint of the chaos itself, a literal physical record of how a chemical reaction happened to unfold inside one specific room, on one specific day, and could never unfold the exact same way twice, even with identical starting genetics. So that solves it. Random chemical noise in the womb, not genetics, decides your fingerprint. Mystery closed. Except there's a strange twist buried inside that explanation, and it has nothing to do with twins at all. The exact mathematical framework Glover's team used to describe those colliding chemical waves wasn't new. It was first written down in 1952 by the mathematician Alan Turing, the same Turing known for cracking Otzi codes and laying the groundwork for modern computing. In a paper called The Chemical Basis of Morphogenesis, Turing proposed using nothing but equations that two competing chemical signals spreading through tissue at different speeds could spontaneously organize themselves into stripes, spots, or spirals, with no outside blueprint required. He had no way to identify which actual molecules might do this in a real animal. He was working 70 years before anyone could test it directly in living tissue. He simply did the math and predicted that biology should be capable of generating exactly this kind of pattern, purely from the logic of the equations themselves. It took until the 2020s for biologists to find real molecules, including the WNT, EDAR, and BMP system behind fingerprints, that behave exactly the way Turing's equation said something should. The same reaction diffusion math now appears to explain zebra stripes, leopard spots, and the spacing of hair follicles, all generated the same way. Two chemicals racing each other across tissue with no master plan deciding the final result in advance. A dead mathematician who never once looked through a microscope at a living cell turned out to be describing your thumbprint 70 years before anyone knew it, which leaves one piece genuinely unresolved. Glover's team could recreate the wave mechanism in mice and explain why patterns settle into the categories they do: loops, swirls, arches. What they still can't do for any individual fetus, human or otherwise, is predict in advance exactly where a specific willy will start or which direction it will curl. The system is fully explained and still completely unpredictable, by its very nature, not because of some missing piece of data scientists just haven't found yet. It's chaotic on purpose, baked directly into the math itself. The same way you can fully understand the physics of a coin flip and still never call heads or tails with certainty before it lands. So the next time you press your thumb onto a screen to unlock your phone, remember what that pattern actually is. Not a code written by your genes, not a decision your body made for a reason. Just the literal frozen shape of a chemical argument that played out inside your mother's womb for a few weeks. One that could never be replayed the same way again. Not even by an identical copy of you, sharing the exact same womb